Talking about DNA, which is everyone's favorite molecule. Yes, you did. We already talked about DNA. You didn't know it was your favorite. But now you do. And I have this laser pointer now, so I can point things without moving. I thought it didn't either, and then I screwed it on a little harder. It works, but it'll probably work better with new batteries. All right, you guys are here now. We can start.
I got you something without batteries.
What? You can't bribe me. Okay, maybe you can. I'll share these later. Yeah. All right, I'm gonna put in these batteries real quick.
By the way, you didn't even know the batteries.
No, I'll pay you back. How much were they?
I bought the races.
Okay.
They weren't really for you, but.
You can have them back.
I'm just kidding. I got to see those anyway.
Oh, well.
I did come in too sometimes.
Okay, so DNA. For a lot of time, they didn't really know what DNA was, and they also didn't know. They knew about genes, or they knew about people inheriting traits before they knew what was causing it. So later we'll talk about Mendel and some of the plants, some of the experiments he did with pea plants. And we'll talk about ways things are inherited from one generation to the next. They knew a lot of that stuff before they knew about DNA. So how traits were passed was a mystery for a while. But they did a few experiments that I'm going to show you that helped deduce and conclude that DNA was the material that was being passed or used for inheritance. So the first one, Frederick Griffith, what he did was he took this strain of pneumonia, which is a bacteria, and he injected it into mice, and with this polysaccharide coat, it would cause mice to die. Okay? They took another strain of pneumonia, and it does not have the polysaccharide coat, so the antibodies or the immune system of the mice was able to eradicate it, and so the mice lived. When they heat killed the pneumonia, so just expose it to heat, so it basically died, and then injected it into the mouse, it lived as well. So in both of these cases, the mouse was just fine. But when they combined the two, the heat killed and the non-virulent, meaning non-lethal, it's not going to kill it. When they combined the two and then injected it in the mouse died. Why? Why? So what do you think's going on here? All right, well, their idea was that something is transforming or something is being passed from this dead one to the alive one, okay? But they didn't know what it was, but something's going on between these two that's causing this live one to become more deadly, okay? So the next guys, Avery, McLeod, and McCarty, they did the same experiment, only they destroyed the proteins in one and they destroyed the DNA in the other one. Okay, so at this point we kind of narrowed it down to it's either the proteins or the DNA that is being passed on from one organism to its progeny. When they destroyed the proteins, everything was the same. So this happened. They destroyed the proteins, did all the experiments, the mice died. When they destroyed the DNA, the mice were just fine. Sorry, I just pressed one. The mice lived. Okay, so what's the conclusion then? Right, so DNA is somehow coming from this heat-killed one into the non-lethal one and making it lethal. Okay, but when they destroy the DNA, everything's fine. Okay, so that's how they narrowed it down to DNA. They did another experiment. This is with Hershey and Chase. It's in the 1952, so it's in the 50s. They looked at these bacteriophages, and bacteriophages are viruses that insert their DNA into bacterias, and then their DNA kind of takes over the enzymes within the nucleus, or not the nucleus, but in the nucleoid of the bacteria, and then it creates a bunch of more of these bacteriophages and then kills the bacteria. So what they did is they radio-labeled some DNA and radio-labeled some protein, and then they let these bacteriophages infect bacteria, and then once they checked the bacteria, they found that the one that was labeled the DNA, they found a lot of the DNA. Okay, but when they labeled the protein, they didn't find any of the radioactive label. So again, this is showing that it must be the DNA that's being passed through the bacteriophage into the bacteria. All right, so that's the conclusion. DNA is what's being passed. All right, what is DNA? Okay, you guys, we already talked about DNA, so you guys tell me what you know about DNA. It's a molecule, good.
It is your genetic code.
Okay, it's your genetic code.
Something double helix.
Double helix, yeah. Your double helix. It's good. Yeah, so it's made of a double helix. What else? Say it again. It's a nucleic acid, right? What's a nucleic acid? Yeah, one of the 4 macromolecules. Yeah. Someone else say something? Okay, what's a nucleic acid made out of? Nucleotides. Good. What's nucleotide made out of?
Protein. Sausages.
No, it's not.
What is it?
Okay, it's made out of a sugar. In DNA, it's made out of a deoxyribose. What'd you say, Steve?
So the mark's supposed to know
Oh, this was on one of the exams.
that? Oh.
Even. A phosphate group and a nitrogenous base. Everyone got it wrong on the exam, though. I think there was only 1 or 2 people that got it right. So. Don't feel bad. Okay, so here is a nucleotide. Here's the phosphate group. Here's the sugar, and there's the base. There's 4 different types of bases in DNA. RNA has a different type. Okay, the 4 different types are adenine, thymine, cytosine, and guanine. And the attachment end is, what makes it deoxyribose is this OH group at the, it says 3', but that should say 2'. Change that to 2'. Okay, if it's a free hydroxyl group right there at the 2', then it's RNA. Oh, wait, no, that's talking about something else. Anyway, the difference between RNA and DNA is just this one molecule. DNA has a hydrogen there, and RNA has a OH there. Both of them have an OH at the 3'. So one thing to note really quick. So this, you probably never heard of this 5', 3'. Does that make any sense to you? Okay, what I'm talking about is each of these angles here, where it comes to an angle, that's where a carbon atom is. And they name them according to where they are. So this is the first carbon. It's 1'. That little apostrophe is called prime. This is the second one. That's the third one. That's the fourth one. That's the fifth one. Okay, and it's just a way to orient the molecule. We don't have a top, bottom, left, right on these molecules. Instead, we're going to talk about the carbons 1, 2, 3, 4, 5'. Yeah, this is just talking about this 3' carbon has the OH. At the 2' carbon, that's what differentiates deoxyribose from ribose, which is the difference between DNA and RNA. So yeah, just leave it at 3'. Okay, the bond formed between nucleotides is called a phosphodiester bond. Okay, and it's a phosphate group bonded between the carbon of one deoxyribose, so one nucleotide, to another nucleotide. Okay, this is the 3' carbon. So if you look back at that other picture, you had 1' here, 2' here, and 3' there. So the 3' of one is attached to the 5' of the other. 1', 2', 3', 4', 5'. Okay, does that make sense? Okay, we're just naming these different angles, which are carbon atoms. We're giving them a name or a number, depending on where they are located next to the oxygen, basically, or in that ring structure. So the chain of nucleotides, so from one to the other, has a 5' to 3' orientation, meaning this one is always attached to, the 3' carbon is always attached to the 5' carbon of the next one. It won't attach to this one or that one or that one. It's only to that one. So they're all in the same orientation. Okay, another guy who did some experiments was Chargaff. Okay, and he looked at the proportion of these bases in DNA. And what he found was the amount of adenine was equal to the amount of thymine, and the amount of guanine was equal to the amount of cytosine. Okay, so this is called Chargaff's rule. So one thing I can do is I can say, you purify DNA in a organism, and you look at the amount of adenine, and it has 17% adenine. Okay, tell me how much thymine, guanine, and cytosine it had.
17%
Okay, so it has 17% thymine. How are you going to figure out how much guanine and cytosine there is? Mm-hmm. So what? 100 minus, you're going to have to add these, so that's 34, 66. And then you're going to have to divide that by 2, which is, so 33% guanine and 33% cytosine. Okay, so I only have to give you one number, and you know the rest. Let's do another one just for fun. So what about, I'll just do an easy one. 40% cytosine.
40% more.
Okay, so we have 40% guanine, because those are always going to be equal. Right, so there's 40, 40 is 80, so there's 20% left, and there's, divide that by 2, you got 10% adenine, 10% thymine. It's a good test question. What's that? Do it again?
How in the world, okay, I see that you're A equals T and U equals C, but how do you get everything else?
Okay, so they're going to be equal. And these are your only 4 options. Okay, so these are always going to add up to 100%. 40, 40, 10, and 10. Okay?
Well, that makes sense. I don't see 100% on there anywhere.
Right. There isn't. All I said was adenine is equal to thymine, guanine is cytosine. But they add up to 100% of the base pairs in DNA. Say again? Yeah, always equal proportion of purines and pyrimidines. So adenine and guanine are purines. Thymine and cytosine are pyrimidines. So I could say, this is another thing you could do. There are, let's say, 70% purines. No, that's just, you just do. So there's 30% pyrimidines. But that's, no, I won't ask that. It's just the different structures. All right, another scientist who did a lot of work with DNA was Rosalind Franklin. She's one of the first women scientists, and that was really kind of the leader or helped kind of breakthrough in her scientific field. And what she did is she determined the shape of DNA, that it's helical in structure. All right? And she was even able to deduce by these, this is one of the pictures she made, X-ray diffraction. I don't know exactly how it works, but able to deduce how tight the helix was, which is, it makes every 2 nanometers, it makes a complete turn. Has a diameter of 2 nanometers, and it makes a complete turn every 3.4 nanometers. All right, Watson and Crick are, they kind of swooped in, and they didn't do any experiments, and then they get credit for basically everything about DNA. So if anyone tells you who figured out what was the who figured out what the structure of DNA was, everyone says Watson and Crick. But all they did was they took all these other, they took Chargaff's rules and the work that Rosalind Franklin did and these other scientists, and they just put it all together and then made a 3D model of what DNA looked like. So kind of cheapskates. They swooped in there and figured it out. But, I mean, no one else had figured it out, so what they did was still pretty cool. And so they figured out that it was a double helix and that the adenines and the guanines and the cytosines and thymines all paired together. I said that wrong. Adenine and thymine, cytosine, guanine. Okay, so then we have a double helix. Not only is it helical, but you have 2 strands of nucleotides that are bound together, and they run in opposite directions, meaning they're anti-parallel. And they're joined together by those phosphodiester bonds between the sugar and phosphate groups. And by opposite direction, so this one you see, again, we have the carbons. Use my laser pointer. The 1 prime carbon is on the inside, and the 3, 4, 5 carbon is on the outside. On the other side, it's upside down. And I have a picture of one in a second. So they're anti-parallel. They're 2 going. They're next to each other, but one's flipped upside down. And they're held together by hydrogen bonds. Where else did we see hydrogen bonds? What else makes hydrogen bonds?
Proteins.
They do. Proteins make hydrogen bonds. What else?
Cells.
Not cells. This over here. Water. Yeah. I was pointing to your Coke, which is lots of sugar and water. But water, right? Water, that's why all the properties of water are due to the hydrogen bonds it makes. And so that's why G bonds with C and A with T, and they don't really bond well when you mix them up because of the structure of their hydrogens and nitrogens and oxygens towards each other, they make these hydrogen bonds. What's nice about hydrogen bonds, they're strong enough to hold them together, but they're weak enough to where they can be pulled apart when needed. Okay, A forms 2 bonds with T, and G forms 3 bonds with C, and that's what gives the double helix a consistent diameter. Okay, so one of the questions they had then, once they figured out what the DNA was the replicating material, was how the DNA was passed on from the parent to the daughter cells. Okay, and the 3 theories were it was either conservative, semi-conservative, or dispersive. And they did a bunch of experiments. We're not going to go over them. But they figured out that it's semi-conservative, which is this one in the middle here. Okay, and just to illustrate that, get up my markers real quick. So they knew that DNA was made of these 2 strands, and they knew that it was copied when you made 2 daughter cells. But they didn't know if, when they made a copy, the second one cell, they didn't know if one cell kept both of them, and then the other cell would have 2 new copies. So this black represents a new copy. Okay, so this would be the conservative model, meaning one of the cells got both of the original copies of DNA, and one of the cells got 2 new copies of it. So this is like when you make a copy in the copy machine. You have your original, you have your copy. Okay, the other one was the semi-conservative model, where when you made these copies, you split them up. One went to each of the cells, and then when the new cell was formed, it also made a copy. So each of them had one of the originals and one of the new copies. And then the dispersive model was just that you had chunks of each in each of them. Okay? All right, so DNA replication, when does that occur in the cell cycle? What phase from this past test that we did?
Yes.
The S phase. Right, yeah. Prophase is when the cell's getting ready to divide, but the S phase is when it replicates. So it happens before it goes into mitosis. Okay? It requires, in order to replicate, 3 things. One, you need something to copy, and right next to something to copy, I want you to write the blueprint. Okay, our parental DNA then is our blueprint. That's the instructions. That's going to tell us how to do what we need to do. Okay, and then you need something to do the copying. Next to that, we have the enzymes, but the machinery. Okay, that's what's going to be doing the work, actually putting the copies together. That's your copy machine. And then you need building blocks to make the copy. That's going to be our nucleotides. Okay, those are our building blocks. And you need those same things to do some other processes called transcription and translation, which we'll go into next. All right, when you break it down, if you have these 3 things, then the processes of each of them, you have to initiate, then you have to elongate or start putting those nucleotides together, and then it has to come to an end, and that's termination. And so we'll go over these in a little more detail, and we'll go over them again when we talk about transcription and translation, because those same steps happen there. Okay, this shows then our anti-parallel strands. Okay, if you look at where the oxygen is, and the 1, 2, 3, 4, 5 prime, 1, 2, 3, 4, 5 prime there, it's upside down towards each other. That's what is meant by anti-parallel. Okay, and the bases are added then to the 3 prime end. Okay, so that's the end that has the 3 prime carbon. So a base would be added on this side here, and it's going to be added on this side here. Okay, so they're in opposite directions. And they're synthesized in the 5 prime to 3 prime direction. So that's just saying the direction of the sugar or the nucleotides is going to be 5 prime to 3 prime. The machinery, the enzyme that does the work, is called DNA polymerase, which adds nucleotides onto that 3 prime end. New question? Okay. So one of the things on here, you'll see the second page. You have the function of the following enzymes. One of those enzymes is DNA polymerase. Okay, and you have 3 of them. There'll be another slide that'll differentiate between the 3. Okay, so don't write anything yet because we're going to tell you the difference. Okay, but one of the jobs of DNA polymerase is to match the DNA bases with complementary bases. So the A's with the T's and the G's with the C's. Okay, and there's 3 different types, but all of them have these common features. They add the new base to the 3 prime end. They synthesize in the 5 prime to 3 prime direction, and they require an RNA primer. Okay, so they can't just add to a naked piece of DNA. It has to have an RNA primer on there to get started. So the way this works in prokaryotes is you have a site called the origin of replication, where everything starts. That's where initiation begins. And it goes in both directions all the way around the circular chromosome until you get to the termination site, and then you have 2 copies of the DNA. It's pretty simple in prokaryotes because they have the circular DNA. Okay? It's a little more complicated in your eukaryotes. Why? What do you know about the DNA of prokaryotes compared to the DNA of eukaryotes? They are, a little bit. What else? Yeah, they're different shapes. Okay, where this one just goes around in a circle and you're done, the eukaryote chromosomes are what shape? What's that? Linear. Yeah, they're lines. They are helical too, but these are helical as well. They're just, you can't see the helix. Okay? All right, here's the 3 polymerases. DNA polymerase 1, it acts on the lagging strand. We'll talk about what that is in a second. And it removes the primers, the RNA primers that are needed. It replaces them with DNA. Right? DNA polymerase 2, it repairs some of the mistakes that are made. And DNA polymerase 3 is the one that's doing most of the work, replicating and adding those nucleotides. Okay, all of them can extract nucleotides when they need to. That's exonuclease activity. So if there's a mistake made, they can go in and take one out, put a new one in. From the 3' to 5' direction. The DNA polymerase 1 also can do it in the opposite direction, from 5' to 3'. Okay, so here you could fill out DNA polymerase 1, 2, and 3 real quick, because you know what they are. Yes?
Did we make one of those?
What's that?
Did we make one of those?
Yes, you did. So that'll be due Tuesday at the end of school, at the end of class on Tuesday night school. Just talk to me after class.
Hmm.
Oh, do you want to take these in?
Mhm.
Yeah, just sit in the back. Okay. You have a few enzymes that, because DNA is in this helical twisting structure, you have to unwind it in order to get other enzymes in there to do their job. So you have a few of these enzymes that are involved in unwinding and rewinding. So helicase unwinds the DNA. Okay, so that's another one you could put. Actually, it's not on there. Oh yeah, it is. Helicase under the DNA polymerase. It unwinds the DNA. The single-stranded binding proteins, they bind to the DNA once it's unwound to keep it from closing back on itself. It needs to stay open for a little bit so that those enzymes can do their job. And then topoisomerase prevents supercoiling. What happens when you unwind, it twists on the back end. That's what supercoiling is. So if you can think of, like, if you've got a rope or a garden hose, when you try and loop that around and then you try and undo it and it's all like still twisted up, what topoisomerase does is it keeps it from getting all twisted up when you unwind it. Okay, so single-strand binding proteins is on there. Topoisomerase is on there. DNA primase is the enzyme that adds the RNA primers. Okay, we'll talk about the other ones in a minute.
On this worksheet, is that third DNA polymerase? I mean, is
Oh
it
yeah,
saying
that's supposed to be DNA polymerase 3. Yeah, sorry. So it says DNA polymerase 1, 2, and 2. It should be 1, 2, and 3. Okay, so because, again, DNA is anti-parallel, two strands and going in opposite directions, and because those enzymes can only work in one direction, replication is semi-discontinuous. What this means is that one strand can go continually as it opens, as the DNA opens, as this figure shows. It opened a little bit there. It can just keep on adding nucleotides as it opens. Okay, so it really only needs to start once, has one RNA primer, and then it just adds these nucleotides. That's called the leading strand, and so it's continuous. The other one is going in the opposite direction, so it's adding nucleotides this way. The problem is it's opening that way, so as it opens, it kind of has to leapfrog down, so it has a bunch of chunks, a bunch of individual strands. Those different strands are called Okazaki fragments. Make sense? No? Okay, man, I should have brought some rope or something. All right, here's my piece of DNA. When I unwind it, okay, now I can add nucleotides. This one I can just start adding nucleotides, okay? Because it's going in this direction. I can add them left to right. Okay, but when I open it up more, I can just keep on adding nucleotides. So that's the leading strand. On this one, though, I have to add them this way. Okay, so I can come over here and I'm starting to add nucleotides this way. And I'm doing just fine. But then when I open it up more, I can't add them on this end. I have to leapfrog over here and add them this way. Does that make more sense? Whereas this one, because I'm adding that way, I can just keep going.
Well, why adds the other way?
Because they're in opposite directions. And my enzymes only work in one direction, so they can only add. So on this strand, the 5' end, the way the sugar's oriented, is the opposite of this one. This one has the 3' end sticking out. And I can add them on only going in this direction. Well, this one's the opposite way, so I can only add them that way. Okay, so it's a semi-discontinuous replication. So where this happens, where these things are being unwound, is called the replication fork. Okay, it's where you're opening or separating those strands of DNA. The DNA primase is an RNA polymerase that makes the RNA primer. Okay, so that's one of the ones on there. So how many primers are going to be needed for the leading strand? The leading strand is the easy one. It's going in the right direction, or going in the same direction as it's opening. So it only needs one primer at the very beginning. Okay? The primer for the lagging strand, you're going to have to put on a lot. Okay? There's no number for it, but you're going to have to put on a lot because every time it needs to start, every time it opens up, it needs a new primer because it has to go in that direction. So every time you open up, well, as many times as it opens up, it needs another primer. Okay, once these strands, the DNA polymerase has finished its fragment, there's still a little piece there that's not bound, and ligase comes in and seals that little piece together, seals the nick. Okay, and then once everything is all done, DNA gyrase unlinks the two copies, so you have two separate pieces of DNA. Okay, so that one's on there too, DNA gyrase. And we've only got one more. Oh no, oh yeah. Only got one more. Okay, this is a figure. This would be great for an exam, something you have to label. Okay, and this is a little more complicated but accurate view of the same thing, the replication fork. So we didn't talk about this clamp loader or a couple of the other things on there, the clamps, but all the other enzymes are on there. Okay, so when you unwind the DNA, which enzyme unwinds the DNA? You can look on your sheet. Helicase, yeah. Okay, so helicase pulls them apart. Now you have a replication fork. But before the DNA polymerase can get started, it needs a primer. What puts the primer on there? Which enzyme puts the primer on? Yeah, primase. So a lot of them are named by what they do. Okay, another thing that helps once it's opened is you have something that binds to the DNA that keeps it from closing back up again. What's that?
No ligase.
Not ligase. What?
The nuts.
It's not an enzyme, but it binds to the DNA to keep it from closing back up.
The long
The long word. SSB binding proteins, yeah. Okay, different long word. All right, and then the different polymerases will add the nucleotides. What keeps it from supercoiling? That topoisomerase. Okay, and then the DNA gyrase pulls apart the strands once it's all done. You got all that? Okay, another fun thing to do is you could draw this out, draw the little things. You have fun with that? Yeah, exactly. On the beach
Every day.
with your whiteboard. Okay, eukaryotic replication is pretty similar, except it's a little more complicated, again, because the chromosomes are linear. And so rather than just having one origin of replication and one termination site, like the prokaryotes, it has multiple replicons. All the enzymes are the same, except eukaryotes have telomerase. That's how I'm going to say it. Telomerase. Okay, and this is just dealing with the very ends of the chromosomes, which are called telomeres. Okay, I mentioned this just a second ago. So a replicon is an area, these are replicons down here, where it has sites which allow the replication to begin. So it has an origin of replication site. Eukaryotes have lots of these, prokaryotes just have one. In order to initiate replication, it's more complicated in eukaryotes as well. We're not going to go into the specifics, but just know it's more complicated. And then the primase includes both DNA and RNA polymerase. All right, so telomeres. We're going to talk about telomerase for a second. Telomeres are the end. So on here it's the red parts of the chromosomes. And they don't have a gene function. They don't express anything. They're really there to keep the chromosome from degrading and maintain the integrity of the chromosome. Oops. The problem is DNA polymerase can't synthesize that last little bit by itself. So these are here, and they're a bunch of basically just long ends, so that every time it's replicated, it'll lose a little bit, but because they're so long and they don't really affect anything, you have a long time before it's going to hurt the chromosome. Good? Okay. All right, so this is just a picture showing that on the lagging strand, where it has the multiple primers, because it's going in the opposite direction, that last little primer can't be, the DNA polymerase can't get in there and put those nucleotides on there. So what happens is you just shorten that end. This is the telomere, so that's the end. Okay, so to prevent that, what you have is telomerase, which is an enzyme that uses a little RNA sequence and a repeating sequence, AT of these nucleotides. And it adds it on there, and then it adds an extension of those base pairs so that you don't have to have those ends chopped off every time. You don't have to worry about this on the leading strand because it's going in the direction it needs to, and it can just go to the end. Okay, so telomerase, the function of that is to, say, prevent the shortening of telomeres. Put that on there. Prevents the shortening of telomeres on the lagging strand. There's no problem in the leading strand.
On the what strand?
Lagging. All right. DNA replication is actually a pretty error-free process, but sometimes errors are made. And there are processes in place to keep errors from being replicated or proofreading. But there are other things that can cause more mistakes to be made, and those are called mutagens. Okay, and anyone familiar with the Teenage Mutant Ninja Turtles know that mutagens will turn you into some sort of animal-human combo, right? Okay, but in reality, usually they just cause cancer. Okay, so radiation and chemicals are, if it's a carcinogen, it's a chemical that causes cancer. It causes mutations in the DNA. Radiation does the same thing. And we know that DNA repair is important because there's lots of mechanisms in place to repair the DNA even when it's damaged. Yeah. So heat will cause damage to cells. What? Tanning beds, yeah, which is radiation and heat. Yeah. Yeah, so don't go tanning unless you want skin cancer. What's that? Okay. Okay, so when something does go wrong, there's two different ways the DNA can be fixed. It can have a specific repair where it targets a specific type of error, or it can be non-specific, meaning it doesn't matter what the error is, it can just fix it. And I'm going to give you an example of a specific one and a non-specific one. Okay, so photo repair is an example of a specific repair mechanism. So this is what happens when you go tanning. Okay, UV light, which is in sunlight or in those tanning bed lights, causes thymine dimers where the thymines bond to each other rather than the adenines across from them. Okay, this is a mistake. You don't want this to happen. So this enzyme, photolyase, what it does is it binds to these areas where there's thymine dimers made, and it keeps the thymine dimers, or it separates them and gets them to bond with where they're supposed to. Okay, this is a specific repair mechanism. It specifically identifies thymine dimers and fixes thymine dimers only. Can't fix anything else. Excision repair, this is non-specific. So it doesn't matter what the error is in the DNA. It just goes in there, cuts it out, and puts in some new DNA nucleotides. Okay, so it's it basically recognizes that there's damage, goes in there, says, okay, this is messed up. We're just going to cut cut the whole thing out. Once it does that, then a new DNA polymerase can go in there and fix it. Yeah. It's science nonfiction. All right, we're going to stop there and take a 5-minute break. This one? Okay, and then we'll do transcription and translation, then we'll be done. But you'll need a blank piece of paper for transcription and translation. Or you can just use the back of this handout. Okay, so I have a few slides that you don't. This is one of them. And this is what you're going to write down on your blank sheet of paper. But you're going to write something more. So just on this top part, write this little part. The central dogma. What's a dogma? You ever heard that word before?
Like a unifying core idea.
Right, a unifying core idea, or like a, yeah. Yeah, they talk about in religion, it would be like your doctrine. Yeah. Okay. The basis on which the church is founded, or whatever. But in science, it's a principle on which other things are based. The central dogma. So the central dogma in biology is this: that DNA is converted to RNA through a process called through a process called transcription. Thank you. And RNA is converted into protein in a process called translation. Okay, so DNA, which is a double helix, there's two anti-parallel strings of nucleotides, is transcribed into RNA, which is also made of nucleotides. So they're made of the same thing. So when you transcribe something, it's like when you're taking notes. When you transcribe something, do you write every single word that, like, do you write every word that's coming out of my mouth? No, what do you write down?
Keyword.
Yeah, the important stuff. When you transcribe DNA to RNA, you don't make an exact copy, you just take the important stuff. Okay? But when you make RNA into protein, these are made of nucleotides. What are proteins made of? Amino acids, right. So it's like a different language. So there you're translating. So that's like taking something from English and putting it into Chinese. Okay? Completely different letters, alphabet, everything's different. Okay? So that's one way to remember it. Another way to remember it is they're alphabetical. So the C, transcription, comes before the L, translation. Okay? Again, this is kind of like meiosis and mitosis. They sound the same, but they're similar processes, but you need to know the differences as well. So transcription, you're going from DNA to RNA. Translation, you're going from RNA to protein. Okay, DNA, the structure of DNA. You guys don't have this either, so break this down. It's made of nucleotides. Nucleotides with bases, including our A, G, C, and T. Okay? You just write down the abbreviations. Okay, and like we said, A pairs with T and G pairs with C. Who was the guy who came up with that rule? Yeah, Chargaff. Chargaff. Okay? In RNA, it's pretty similar, but just slight difference. Instead of thymine, you have uracil. So A pairs with U and G pairs with C again. And now you have a different nucleotide or different base. Okay, and so DNA is the blueprint for our RNA. RNA is the code by which we can code for amino acids. All right? There's 20 different kinds of amino acids. Each of them have slightly different molecular properties. Those molecular properties are the basis by which you can put them together and make proteins. And so what happens is you have codons, which are 3 RNA nucleotides, will code for our amino acid sequence. And that's how you make your protein. Okay, these are the basics of our central dogma. You have to know what DNA, RNA, and protein are made out of. And then we're going to fill in the gaps of how the process takes place. Okay, a lot of this, though, we've already talked about. We talked about DNA. We haven't talked about RNA, so we'll talk about that first. And we've talked about protein. We talked about macromolecules. And where is all this taking place in the cell? In the nucleus, right? That's where our DNA is. So transcription takes place in the nucleus. That's where you make DNA into RNA. RNA is then taken out of the nucleus. And what is the organelle where proteins are synthesized? Not Golgi apparatus. No. Rough. Endoplasm. What's the rough for? Ribosome. So ribosomes. Oh, I didn't hear it. Say it louder. Muffled. Okay? So the process of translation happens at the ribosome. All right, so remember we needed a blueprint, machinery, and building blocks. We needed those for what process? Replication. What's that? You don't have this, so you'll need to write this down too. Because I wanted you to write them down. One of the things, so I don't make you write down everything, but sometimes it's better to write it all down. Helps you get it in your head a little bit better. Yep, I do. So, in the processes of transcription and translation, they all need a blueprint. They need some instructions. They all need something to do the work, which are our enzymes, and they all need building blocks, something to put together. It's like building a cabinet. When you buy something from IKEA, anybody ever bought something from IKEA? With the pictures for instructions? Anyway. You have to have all the pieces there, okay? Those are your building blocks. You have to have some tools, like a screwdriver, and your hands. You have to be thinking clearly. And you need a blueprint, so you need those instructions. If you don't have the instructions and you try and put it together, what happens? You put it together, and then you have all these extra pieces. You're like, or you start to put it together and you put something on backwards, then you have to take it all apart. Right? Okay. Then the process. In transcription, the blueprint is the DNA. That's our instructions. Specifically, the template strand. Okay, there's 2 strands on the DNA. One of them is called the template strand. Our enzyme that does most of the work is RNA polymerase. Okay, so DNA polymerase was the enzyme that did most of the work in replication. RNA polymerase is in transcription. And our nucleotides with the 4 different bases. I would just abbreviate those A, G, C, and U. Okay, and the process involves initiation, elongation, and termination. These are the same as in replication. You got to get it started. And you got to start adding things, elongating. And then once you're done, you got to terminate it. You got to stop. So, you guys can probably figure out the rest without maybe going through it. What's the blueprint in translation? RNA, yeah. Specifically messenger RNA, mRNA. Yeah, we haven't talked about the enzyme yet, so you probably don't know that, but the enzyme, the guy that's doing the work, is called, the ribosome. We'll talk about that, which is made of rRNA, ribosomal RNA. And then the amino acids are the building blocks, and they are brought to the site of translation by a tRNA, or a transfer RNA. And then guess what the process is? Initiation, elongation, termination. Okay, again, we're kind of putting the big picture, the basics. We're going to fill in the details. Okay, this is a good thing to know. A lot of good test questions on here. Yep. Oh, you got it all?
No.
You have half of it? Craig's smart. He just took a picture. You can take a picture if you want. Okay. Do a selfie. Yeah.
Can you go to that? What's in the
This one? All right, while you're doing that, here's your chance to be on camera. Yeah, now you'll watch it. It's probably gonna go viral now. I should put the title like, "You will not believe what happens during this biology class." Or something. Okay, we got everything? All right, you guys have this. So this is just saying there are different types of DNA, RNA, and we already talked about a couple of them: messenger RNA, ribosomal RNA, and transfer RNA. We're going to mention these other ones, but I want you to know specifically those three, because those are very important for transcription and translation, and the test. Yes. Okay, messenger RNA is the template for translation, and it's what we make in transcription. And it's not a double helix, it's just one strand. It doesn't really take any form. Ribosomes are made of ribosomal RNA, and transfer RNA have amino acids on it, and those are used for translation as well. Okay, so Francis Crick and Sidney Brenner, so we got some more scientists if you'll notice on here, there's some matching on the second to last page. I think that's the third page. That's the back of the second page. Has all the scientists there for you, and then what they did, so you can match them up. So it doesn't have Crick and Brenner on there though, just the other ones. Okay, but they figured out that you can take 3 RNAs or 3 nucleotides of DNA, and they will code for an RNA, and then those RNAs will code for different amino acids. Okay, and the way they did this was they would take a strand of DNA and they would insert a nucleotide, or they would take one out and figure out how it changed the amino acids. Okay, so on this one they deleted these 3 bases, ACG, and they noticed that the amino acid that was made didn't have this threonine on there. So then they determined that that ACG coded for threonine, which is an amino acid. Okay, they did the same thing here. They deleted a base, and so now instead of it being an ACG, it was an AGC. So they shifted over. Now they had serine instead of threonine. Okay, so this indicates that the importance of the reading frame. So basically, you can't take one out or put one in without messing up this whole sequence and then messing up the amino acids it codes for. Okay, another scientist, Marshall Nierenberg, he identified specific codons, including the stop codons. There's 3 of them, UUA, UGA, and UAG. Okay, and I remember learning some acronyms for them, but I can't remember what they are. It was like, the University of Georgia is awful. University of Alabama's gross, or something. Something. I can't remember. You don't have to know them. They'll be given to you. Another one, start codon, is AUG. So anytime there's AUG, that's going to be the first codon, which also starts codes for the amino acid methionine. And then the rest of them just code for different amino acids. Okay, the code is degenerate, which means that some codes can code for the same amino acid. Okay, more than one codon can code for the same amino acid. And it's practically universal, meaning almost every living thing uses the same sequence of codons for the same amino acids. So you, plants, bacteria, you're all made of the same amino acids. They're just in different orders, different lengths. Okay, so these are all the different amino acids, and these are all the different codes. So one of the things you're going to have to do is figure out, let me see if I have this on here, nope. Figure out what codes for what amino acid. So, if I put 3 letters on here, AUG, what amino acid does that code for? You can use this table to find the first letter, which is A. So you go to the A. Then the second letter is across the top. Our second letter is U. And then you look in that box to find the one you need. So AUG codes for methionine or start. Okay, I'm going to give you another one. UUC. What does that code for? UUC. Phenylalanine. Okay, good. All right. How about GGG? G, G, G. Oh, I see. Good. So, and you can see here that all of these codons code for the same amino acid. So that's what we just talked about. You don't have to know all of them. If I ask you to do this, I will give you this table. But you just have to know how to do it. UUA. Lucy. Good. Speed round. Lightning round. Yes.
Do you need to have it, you can like map it up or what?
Yeah. So yeah, if I give you, I'll show you in a second, but and one of the questions on here asks you to do this. On the last page, there's a string of amino acids here. Okay, and this is a DNA strand. Okay, so if this is going to code for a protein, what does it have to be converted into first? It's DNA. Has to be transcribed into RNA. So one of the things you're going to do is match it with the base pair. So A binds with what base pair of RNA? No, that's DNA. U, right? So you put a U under the A. T binds with A. Yep. And so you got U, A, U. C binds with G. And then G binds with C. Right. So you go all the way down and you fill that in. And then to start translation, what do you need? AUG. So then you have to go along that thing you just made and find the first AUG, and then that's where you're going to start. So AUG, that'll be methionine, and then the next triplet will be, I don't know, and then you'll match it up with the next one, so on and so forth, until you get to what? One of these. So if it's AUG and then the next one is UAA, you would stop right there. You wouldn't have to go any further. Because that's one of the stop codons. There's 3 stop codons. If you run into any of those 3, then that's where it would end. And we'll practice this in lab too.
So,
Yeah? Okay. All right, so how this works now, these are the details. You have just one RNA polymerase, so then this is the machinery doing the work in transcription. Doesn't require a primer. It can start all by itself, goes in there, unzips it, starts putting in nucleotides. It does require a promoter, which is a region in the DNA that it recognizes. And then a start site, which is another region in the DNA that it recognizes to start. And then it needs to end at some point, so it needs a termination site. Okay, the promoter is the region it recognizes, again, to bind. Recognizes and has a binding site for the RNA polymerase. And then is found upstream of the start site. So we have a start site right here. Upstream to that is going to be the promoter. And it's asymmetrical, meaning it's only going to go in one direction. Because if it went the other direction because it's opposite, it would be creating something different. Elongation then, it starts adding nucleotides on the end of these RNAs. Once it opens up, starts making RNA nucleotides and adding them together. The place where that is occurring is called the transcription bubble. And as it moves along, it opens up on one end and it closes it behind itself. Okay, it also goes in the 5' to 3' direction. Again, that's just talking about the direction or the orientation of the RNA nucleotides. Termination, once it hits the termination site, that will be the signal for it to stop transcribing. The RNA will make one of these hairpin loops. Okay, the hairpin loops allow the RNA polymerase to let go. The DNA rewinds itself, and then now you have a messenger RNA. Did you miss one? The bubble? Hairpin? Got all those? In prokaryotes, you don't have to wait till translation is done before transcription is done before you can begin translation. So you have transcription and translation going on at the same time. The operon is a sequence of DNA on the DNA that allows the turning off or turning on of transcription or translation.
Can you go back one slide, please? All right, thank you.
Got all these. Okay, in eukaryote transcription, you have 3 different RNA polymerases. RNA polymerase 1 transcribes the RNA. RNA polymerase 2 transcribes mRNA, and RNA polymerase 3 transcribes tRNA. And we already discussed their functions. Each RNA has its own different promoter that it recognizes. That's how it can keep from binding to the wrong spot. Eukaryotic transcription is a little more complicated. It requires all these transcription factors, which are proteins that also have to bind to the site before the polymerase can bind and start transcription. So it's a big old complex of proteins rather than just one protein doing all the work. And the termination sites are not as well defined. Okay, when these transcription factors and the RNA polymerase bind together, that's called the initiation complex. Okay, so that's what we have here. Transcription factor, a bunch of other transcription factors, they all have to be arranged and binding to the right spot. And once you get your RNA polymerase, it forms your initiation complex. My pointer go. Okay, before the mRNA, this is in eukaryotes, can be used for translation, it has to have a couple modifications. So that it's given a poly-A tail and a cap. And that just helps to orient it and to protect it from being degraded. So if it's just left at a free end, it may bind to something that's not supposed to, or it may just start losing nucleotides. And then not even all of the RNA is used. So some of the RNA is taken out. Those are the introns. Then the only thing that is used is called the exons. And the spliceosome is the complex of proteins that takes the introns out and keeps the exons. And the exons can be rearranged into different ways to create different proteins. And so that's how well, I'll mention this in the next slide, but that's how they originally thought that it was going to be close to 100,000 genes to code for all the proteins in the human genome, but turns out it was closer to 20,000. The reason that is because you can make different proteins from the same gene just by mixing up the exons in a different arrangement. The small ribonucleoprotein particles, or SNRPs, they're the ones that recognize the boundaries between the introns and exons. And when they're recognized within the spliceosome, you can use those to take out the exons. Okay, I'm trying to get through this for you guys. Everyone got that? Okay, this is what I just said. When you have different exons in different arrangements, that's called alternative splicing. Which is how 25,000 genes code for 80,000 different mRNAs. You can mix them up. Got those? Okay, tRNA, its function is to take the amino acid to the ribosome, and then the ribosome binds those amino acids together. The way that it recognizes which amino acid to be there is through a part called the anticodon loop, and that hydrogen bonds with the mRNA. The enzyme that adds them together is called aminoacyl tRNA synthetase. Don't worry about knowing that. Helpful. Okay, everyone got this? Okay, each one of those enzymes recognizes one amino acid, but several tRNAs again, because that's degenerate. A charged tRNA is one that has an amino acid and an ATP, so it's ready to use that energy. The ribosome is ready to use that energy to bind those amino acids together. But there can be mistakes made because ribosomes do not verify the amino acid. They just recognize the anticodon, bind them together. Good? Okay, so the way translation works, ribosome has 3 different sites in it, the P site, A site, E site. The P site is what binds to the tRNA to the growing peptide chain. The A site binds the next amino acid, and the E site is where it links. So put them together, you have 8. Exit, entrance, that's where the work is going on in the P site. So the 2 functions of the ribosome, they decode the RNA and they form peptide bonds. And the peptidyltransferase is the enzyme component, and that's what forms the peptide bonds. So remember when we talked about macromolecules, peptide bonds are what hold together amino acids. Okay, got next. I got 10 more minutes. Do this. So in prokaryotes, the initiation complex has your charged tRNA with methionine, but it's a different type. It's called N-formyl methionine. The small ribosomal unit comes together with the messenger RNA. The first tRNA that has this formyl methionine on it then binds to the mRNA and the small unit. And then the large subunit comes, and now you are ready to elongate. So that's initiation. In eukaryotes, it's very similar, except rather than that big long one, it's just methionine, which we talked about before. The initiation complex is a little more complicated, and it does not have RBS, which is the unit that binds to the 5' cap of the mRNA. You good? Elongation, you just get this tRNA goes there and it goes out. This one moves over to the P site, and a new one comes in with a new amino acid. Then it binds them together. Then this one moves over. And so basically the ribosome moves down, new tRNAs come in, and as it does so, it binds the amino acids together. And so you get a bunch of amino acids bound together. Okay, this is the cycle. Okay, and we already talked about this. The reason why it's you can have multiple codons for the same mRNA is because this last one doesn't have to pair perfectly. So sometimes that third one, it doesn't matter what's there. It's just the first two that matter. So you don't need as many tRNAs for all the codons. Got it? Termination, when you hit one of the stop codons, it initiates some release factors, and then the polypeptide, which is your protein, is released, and the ribosome falls apart, and the messenger RNA is released. All right, we'll do the mutation when we get back. Yeah, but it's kind of hard to talk about, so I can't speed through it. Okay, everyone say goodbye. Have a good spring break. Look for yourself on